Hello everyone. Thank you for joining me today. I am excited to take you on a journey through the captivating history of microbiology and immunology. Today, we'll explore the milestones and discoveries that have defined these fields. Let's dive right in. Our journey begins with Anton van Leeuwenhoek's discovery of microorganisms in 1673. Moving to 1796, we'll delve into Edward Jenner's pioneering smallpox vaccine. The 19th century showcases Louis Pasteur's revolutionary contributions to the theory of biogenesis and anthrax vaccines. Robert Koch's establishment of Koch's postulates in the same century marked a crucial step in disease identification. In the 20th century, witness Alexander Fleming's discovery of penicillin. Addressing contemporary challenges, we'll discuss antimicrobial resistance. Our conclusion will reflect on these milestones and their lasting impact on public health. Let's begin. Now, before we delve into the historical milestones, let's take a moment to understand the profound significance of studying microbiology and immunology. These closely related fields are the cornerstones of our understanding of infectious diseases and the body's defense mechanisms. Microbiology allows us to explore the microscopic world of bacteria, viruses, and fungi, unveiling insights that shape medical treatments, diagnostics, and public health strategies. Immunology, on the other hand, unravels the complexities of our immune system, laying the groundwork for the development of vaccines and immunotherapies. Together, these disciplines contribute to the advancement of medicine and the protection of global health. Our journey begins in the 17th century, when a game-changing tool emerged, the microscope. Invented by Anton van Leeuwenhoek, this revolutionary instrument allowed scientists to explore the previously unseen world of microorganisms, opening up a realm of possibilities in the study of biology. Leeuwenhoek observed saliva, lake water, and rainwater with his single-lens microscope and first discovered microorganisms such as unicellular algae, protozoa, and bacteria. In 1673, he shared his initial findings with the Royal Society of London. Leeuwenhoek's keen observations of microbes laid the foundation for what we now know as microbiology. Shifting gears to immunology, Edward Jenner developed the smallpox vaccine in the late 18th century. The image on the right shows Jenner performing his first vaccination on an 8-year-old boy named James Phipps. In 1796, Jenner observed that milkmaids were immune to smallpox after contracting cow smallpox, or cowpox. He then inoculated Phipps with pus collected from Sarah Nelms, a woman who had previously caught cowpox. Phipps was successfully prevented from contracting smallpox. Subsequently, Jenner developed a vaccination method to prevent smallpox by inoculating people with cowpox. Jenner's vaccination marked the beginning of immunization and the fight against infectious diseases. Fast forward to the 19th century, where Louis Pasteur's groundbreaking work disproved spontaneous generation and introduced pasteurization. Aristotle, an ancient Greek philosopher, insisted on the theory of spontaneous generation. This theory posits that living things can accidentally arise from non-living things such as soil or rotten wood. The debate over the spontaneous generation of living things had persisted for 200 years since the 1600s. Pasteur conclusively ended the debate in 1861 by publishing the book
Sur les corpuscules organisés qui existent dans l'atmosphère. Examen de la doctrine des générations spontanées.
In this book, Pasteur conducted an experiment using meat broth and a swan neck flask, as depicted in the figure. First, he poured meat broth into the flask. Then, he heated the neck of the flask into a narrow S shape, giving rise to its name, the swan neck flask. In the swan neck flask, Pasteur boiled the meat broth. Dust or microorganisms were trapped in the S-shaped glass tube wall of the flask. As a result, when this broth was cooled and left untouched, microorganisms did not form. However, if the neck was broken, it led to the contamination of the flasks, facilitating the growth of microbes within the broth. Through this experiment, Pasteur refuted the controversial theory of spontaneous generation and established the theory of biogenesis. The theory of biogenesis asserts that living things can only arise from existing living things. Pasteur also developed pasteurization methods, anthrax vaccines, and rabies vaccines, laying the foundation for preventing infectious diseases. In 1881, Pasteur demonstrated the efficacy of the anthrax vaccine in front of the public, through an experiment using his own anthrax vaccine on sheep. Here are the details of the experiment. In 1881, Pasteur was provided with dozens of animals for the public trial of the vaccine at Paola-le-Fort. He first divided 48 healthy sheep into two groups, labeling them Group A and Group B for convenience. 24 were assigned to Group A, while the remaining 24 were assigned to Group B. On the 5th of May, 24 sheep in Group B were injected with an anthrax vaccine. On the 17th of May, the animals in Group B were again injected with the vaccine. The sheep in Group A were not vaccinated on both days. On the 31st of May, both groups received injections of virulent Bacillus anthracis, the bacterium that causes anthrax. The experiment drew wide attention. When the condition of the sheep was checked on the 2nd of June, all the vaccinated sheep in Group B were healthy. In contrast, all but 4 of the unvaccinated sheep in Group A were dead. The remaining 4 were also in poor health. In 1885, Pasteur created a vaccine for rabies as well. His contributions revolutionized our understanding of infectious diseases. Another microbiologist named Robert Koch emerged in the late 19th century. Koch discovered the bacteria of anthrax, tuberculosis, and cholera. The image on the right shows a photomicrograph of Bacillus anthracis, the bacterium that causes anthrax. Perhaps more importantly, Koch devised a method of culturing and studying bacteria, thereby establishing the process of identifying the causes of infectious diseases. This systematic process was established as Koch's postulates, and the 4 criteria are as follows. 1. The microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. 2. The microorganism must be isolated from a diseased organism and grown in pure culture. 3. The cultured microorganism should cause disease when introduced into a healthy organism. 4. The microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original specific causative agent. In sum, the works of Koch marked a crucial step forward in disease diagnosis and understanding. This is why he is often honored as the father of bacteriology, alongside Louis Pasteur. The 20th century brought us Alexander Fleming, whose accidental discovery of penicillin initiated a new era in medicine. In 1928, while culturing Staphylococcus, a type of bacteria, Fleming noticed something unexpected. The image on the right is a scanning electron micrograph of Staphylococcus with false color added. Penicillium, a type of fungus, accidentally flew into the bacterial culture medium. Fleming observed that bacteria could not grow around the penicillium. He then extracted penicillin, a substance that inhibits the proliferation of bacteria, from penicillium. The image on the left displays penicillium, and the one on the right depicts the chemical structure of the penicillin core. Soon after, mass production of penicillin became possible, saving many people's lives. Antibiotics, such as penicillin, are substances used to treat bacterial infections by killing bacteria or inhibiting their proliferation. The advent of antibiotics revolutionized our ability to combat infectious diseases. The progress made is impressive, but challenges, particularly antimicrobial resistance, need attention. Antimicrobial resistance occurs when microorganisms evolve to resist drugs designed to combat infections. Antibiotic resistance, specifically, refers to the ability of bacteria to resist the effects of antibiotics. Here's a breakdown of how antibiotic resistance develops. Initially, most germs are drug susceptible, with only a few being drug resistant. Antibiotics kill bacteria that cause diseases, but they also affect some beneficial bacteria. The drug-resistant bacteria that survive multiply, and some undergo spontaneous mutations that make them even more resistant to the antibiotics' effects. Even worse, genetic material can be shared through mechanisms like horizontal gene transfer, allowing resistance genes to spread among different bacterial species. Inappropriate use of antibiotics, such as not completing a prescribed course or using them unnecessarily, fosters an environment where resistant bacteria survive and thrive. Overprescribing antibiotics for viral infections, which antibiotics cannot treat, also contributes to the development of resistance. Safeguarding against this challenge requires responsible antibiotic use in healthcare, surveillance of resistance patterns, and public awareness promoting prudent antibiotic practices. In conclusion, the history of microbiology and immunology is a captivating journey marked by scientific breakthroughs, medical innovations, and an ongoing pursuit of understanding and conquering infectious diseases. These fields remain at the forefront of public health. Thank you all for your time and engagement. If you have any further inquiries, please don't hesitate to reach out in the comments below. Have a wonderful day. Thanks for watching. Don't forget to like and subscribe.